Hello. Hello.
Hello.
Yeah, but are you able to hear me?
Yes, Doctor. Shall I share the presentation from my side?
You once more, then if it will not work, then you can. Is it okay?
Is it okay now? I'm sharing the screen. You can start.
There is too much disturbance. I don't know why. Yeah. Hello. Hello.
Yes, sir.
Yeah. Am I audible? Am I audible?
Yes, doctor. Yes, doctor. You can start.
Okay. Yeah. Good morning, everybody. Today, the topic of presentation is the herbal grassroots formulation for insect pest problem in agriculture. This is, I think it is 12th day. You people must be aware with this stage activity, what stage is doing, and what kind of practices, grassroots practices we have. You must would have come to know about that. I will use some of the similar things in the mainstream of science, how people is working on such kind of knowledge to decipher the science. This is one of the paper which is showing about the formulations of pesticides from some of the medicinal plant. Looking some problem. These are the plants which was used in the formulation, that is Tridex, procumbens, Buddleia, common scientific name is Buddleia chinensis. This is another example which is from our lab, from SISTI lab, that is regarding the formulation of herbal pesticides for the insect pests from some of the grassroots practices. This map is showing the practices which we have documented from different parts of the Gujarat. I'm facing some problems, some disturbances. This is one of the information related to patent on the grassroots practices. This is herbal formulation for control of agriculture pest. This is depicting the plants which is mentioned in the patent and their compositions. This is again the practices of NIF. So this is the composition of the formulations, that is grassroots practices. You are looking the name of the plant, Nerium, Nigella, Annona, then Cucumis, Ancostema, Dedicta. These are in different concentration. A formulation is made by grassroots people, and after validation, we have found the positive result. Then we have filed the patent and documented, and then this is the information on the patent of that particular practices. And this is the formulation of that practice. This is controlling for the insect pest, particularly for the fruit and vegetables. Through this slide, I would like to show you how we can look the some of the practices from the SRISTI database. In coming slide, I will show you these sites how to go to the particular library site of SRISTI database to look into the practices. Here, if you open this page of the SRISTI on the SRISTI website, that is particularly the medicinal plant database of SRISTI, and this on the search icon, if you put any name of the disease or the pest and then search, you will be able to see the different kinds of practices which is mentioned in the honeybee database. You will come to know about that. I am showing you some of the example of the practices with their innovators. This practices is from the Bihar, and that is for the control of larval pest, sucking pest and the larval pest. And this formulation was made in the Bihar, and when we have from this lady, when we have validated these practices in our lab, we have found a positive results. This is another practice from the Gujarat for the control of smut disease in sorghums. And again, the plants used were Aristolochia bracteata. This is commonly known as kidamari. Kidamari means killing the kida, that means the insect. The name of the plant itself is denoting its medicinal property. Another plant which is used by this innovator was chlorhodendron. When we have tested this formulation for the smut disease in vitro condition, we have found a positive results. This is another formulation for the controlling of stem borer. Borer is a very severe problem in different crops, particularly even the vegetables, also particularly the brinjals and the lady's finger, etc. So here this innovator has used a plant named wood apple, that is Limonium acidissima, along with the leaves of the neem plant, Directa and onion. When after validation, we have found the positive result of these practices too. This is yet another practices for leaf curl. This practices has also shown a very good result, and the plant used by commonly known as a soap plant in the local language, that is the Balanites aegyptica. This plant is having very good content of saponin. This was also found controlling in addition to the leaf curl, which is basically a vector-borne disease of virus, but this plant is able to control the vectors. That's why the leaf curl is getting control. In validation, this has also given a very good result. This is another plant which is commonly known as saru, that is the Casuarina equisetifolia. And this plant in the vegetable pest, it has shown the good results. The why I am showing you this slide to make you aware how we are coming to know about the name of the plant which is being used by different innovator for different purposes. Next. Yeah. This is for the control of stem borer in paddy, and the plant being used was acacia, a very common plant, and then apomia, that is commonly known as daffodil. This is yet another practices to control the termites. Next. And the plant used was commonly known as boeringni, and the scientific name is Solanum suratens. Next. See, these are the sites which is mentioned here. If you just click on these sites, you will able to see the page which I have shown you earlier for the searching of the different practices. And if you put your keyword, whether it is related to wilt or the any kind of fungal disease or bacterial disease or any kind of pest, whether it is the pest means the insect, whether it is the hemipteran, dipteran, coleopteran, whatever insect it may be, or even you may put the aphids, acid, or the white fly, you will able to see the different practices which is available on the common database of the honeybee network. Next. See, this, here I am showing you the with the keywords, whatever this is the name of the plant which I have found on our database and what for they were used. These are the particularly the all the plants being used for medicinal purposes can be said as a medicinal plant, but it has the name here which has been mentioned here is having the role in control of pests and diseases in the agriculture. That's why the a list is being prepared from our database. Next. These are the number of practices which is available currently in our database for this particular kinds of diseases. So for example, damping of seedling rotting and many other, and this is number at the right side indicating this much number of practices is available in our database for that particular practices. So whatever problem you would like to search for in our database, you will come to know a lot of information. Now the question is, these are available in the raw form. Raw form means the way innovator has depicted the more information to the scouts. Scouts have written it and came to the came to the SISTI office. There they have documented it and put in a database. Laboratories, particularly SISTI laboratories' job is to do the validations. Whatever practices earlier I have shown you, that was already validated and a positive result was found. Similarly, many of the practices are available, more than 50,000 practices are in total are available, and they can be categorized and searched from our database. And this is the pathway how to, from a grassroots practices, we can come to the formulation of a product. So the first step in the validation or the product development from the grassroots practices is the identification of plant material. Then after procurement, then its processing as per the innovators practices or scientific way in which it can be processed. There are different ways of processing for the botanical material depending on the nature, how the innovator have done that. We need to do the processing for that practices to get the similar kinds of result. Then our job is to put the quality control, because innovator is an informal person. They are may or may not be aware with the science, but here when we are doing the things in the lab for the product development, we need to put the quality control aspect so that batch to batch formulation or batch to batch product development would have the similar results. So first thing is the identification of the plant, then it's quality checking for the from the taxonomical point of view as well as from the biochemical point of view. For biochemical point of view, we do the extraction. In earlier lecture, you would have come to know about what are the different methods of extraction and how we are doing the extraction. I'm not going in detail of that. And then how we can store the extract in the laboratory for the further processings. Then it's formulation. Formulation is a new word, particularly for the main common stream people, because this is a basically the students who are from the pharmacy background, they are familiar with the term formulation. Formulation is nothing but the mixing with the different ingredients along with the active ingredients. And these ingredients may be the compound which is responsible for protecting the active ingredient of the compound to increase their shelf life, to maintain their shelf life, or even to enhance their particular efficacy. So in formulation, basically we are mixing three things. one and foremost thing is to add the some material which is protecting it from the spoilage, that is from the disintegration of the material. So some stabilizer is mixed with the main active ingredient, and then because the active ingredient concentration is less means if we suppose it is the 1 gram or 1 mg, and we need to formulate suppose 1 liter, that means 1 mg per liter is 1 ppm. If the practices is effective at 10 ppm or 100 ppm, accordingly we can do the dilution of the extract for the formulations. So for the dilution, if the extract is in the aqueous, then no problem, we can mix the water. But if the extraction is any organic solvents, then you need to some carrier. Water may not be suitable for that. Depending upon that, there are different nature of the formulation. That means the one is the oil into water and or the water into oil. So we need to develop the emulsion sort of things, because if even there are several practices where oil is being used for the formulation, even the neem oil or the currant oil or the castor oil, if you mix the oil in water, it won't mix. But if you do the saponification is the mixing of the fat with the alkali. That is the normal process of soap formation. Accordingly, different emulsifier is being made by different company, which is being used in the formulations. So you need to select different emulsifiers for your particular active ingredient if it is in the not, if the extract is not in the waters. And you may add some additional ingredient which is able to increase the efficacy. That means having the plant which is having the synergistic effect with the main plant of the practices. Then again, after developing the formulation, you need to check the stability at the room temperature for short duration, for long duration, at least for 6 months to 1 year, to know the efficacy of the formulation and the disintegration coefficient of the formulation. And accordingly, you can establish, because the formulation, all the formulation is of plant-based. So you may develop the either the band pattern for the secondary metabolites or even some primary metabolite if it is having that. So for example, some proteins may give result, but you need to identify from the mechanism of action point of view whether the result, whatever we are getting for the control, is from the primary metabolite or the secondary metabolite. So accordingly, we need to develop the band pattern from the HPTLC, or even if it is the either any different kinds of carbohydrate, protein, or fat. Accordingly, we need to identify the marker compound of that particular compound, which is giving the efficacy or the result. Once that has been established, then you can in subsequent batch of formulation, you can look for that quality and quantity point of view of that particular band pattern in the HPTLC to maintain the formulations. Then of course the you can do the bottling, labeling, and storage for the dispatch with or without secondary packing. This is in nutshell the way how the formulation after validation is being developed in the form of products. Next. These are the sum of the product where we have, these are the peaks which we are looking, is showing the marker compound of different products which we have developed at system. Next. This is, as I told you, we may develop the for the different primary and secondary different markers. These are the markers in one of the plant which we have given the number, code number 60. These are the different band patterns which are looking. Next, and these are the different solvents. Suppose OH, MUH, these are the different solvents in which the extraction is being made. Again, these are the name of the plants is mentioned here. In this particular, we are mentioning the efficacy for the control of different pathogens, because many of the practices for the fungal control or the bacterial controls, if we do the extraction in aqueous either hot or cold or even in the different non-polar solvents, the efficacy varies. Even the compounds concentration varies. So that means depending upon the solubility in different solvent, we can do the extraction, and after that, when we have checked the efficacy by means of plate assay, and these are the highlighted one is showing the higher efficacy of the control for different pathogens. The pathogen which we have tested was E. coli, then the this SRES, and then Pseudomonas, Bacillus, even the Fusarium and Astylagus. Next, again the same in different plants. The purpose why I am showing you these slides will discuss in next slides. Next. These are the plates which is showing the control of Fusarium and Astylago by balanites. Next, yeah, again same thing to visualize the how it looks like. Once you will get a zone size of more than 20 mm at least, only then you can consider the extract of having some potential to be formulated in the form of products, because you may get 1 mm zone size or 2 mm size, but that is not good enough to develop the formulation. So we need to have to cross certain limits, and as per literature in some of the papers you will find at least we should have a efficacy of 20 mm zone size to be developed that particular plant in the form of different formulation to control the pathogens. Next. Yeah, this slide is itself showing is the importance of this kind of work. There are different products in the markets. Whether we can develop similar products from our grassroots practices of such nature. The first paragraph is giving the example of one Nobel laureate who has developed antibiotics for the plant, that is milbemycin. It is a macrolides, and having the this is the chemical structure ivermectin. He was also the one of the Nobel laureate of 2015 for this discovery. Similarly, ivermectins, milbemycins have attracted considerable attention and have been developed as acaricides. Acaricides is a class of pests, that is the insects, which is, belongs to acarids class, and that is having the 6 legs. Then the insecticides and anthelmintics. Anthelmintics with those which belong to helminth and nematode class of nematode class. So the people who have milbemycin is basically a product of microbial origins, and that microbes was found from the isolated from soil. We are having a lot of agriculture practices from which we have isolated even the many microbes, and many plant is having the antimicrobial activity, particularly for the plant pathogens. We can develop a compound similar to milbemycin natures, or even the most highly effective pesticide currently in the market is commonly known as coragen. Coragen is basically a protectant rather than an control agents, and people are this product is having such demands in the market that people are even doing the black marketing of that. The purpose of telling these things is that since milbemycin is originally discovered by a Japanese, and it is having the origin from the plants, we may develop a similar kind of compound from different our grassroots practices if we are intended to. We have developed some of the compounds from our lab for different purposes, and one among them was the basically the antibiotics similar to the amikacins. Similarly, we have developed the from the plant of which is controlling the pesticide. From them also we have identified one compound which has been identified. Likewise, we are the database, 60 database is having very high potential to develop a new class of compounds, which is as per the current norms of government regulation for the development of environment friendly, environmental sustainable pest and disease controlling agents. Next. Same thing. Next. This is particularly based on the mechanism of how the insect and this plant interactions help in the controlling of the disease. If we can add the enzyme chitinase, because all the insects have a cuticular layer of chitin, if the plant along with the interaction during the interaction of the insect can release or secrete the enzyme chitinase, that can help in controlling the different pests. These are the based on the plant microbe or the plant insect interaction point of view. Scientists are on the way, particularly the plant biotechnologists, to develop a transgenic for the chitinase, or even the formulations from the chitinase degrading microbes. Different oligochitin are in the market to just giving the stimulant kind of thing to existing phyllosphere organism to produce the chitinase to control the particularly dissolve the chitin of the insect. But these formulations are, or even the product which is in the market, are at a very primitive stage, and our database is a very good resources to give a direction for this kind of research to develop a new formulation from that point of view. Next. These are the examples which I have shown you the first the photographs. The same photographs is here for controlling different pests. That is sucking pest, chewing pest, then mites, termites. And this another one, though this is not the topic, but these are the varieties which is being developed by the grassroots peoples, and that is in the market. Next. And we are having a company at SISTI named SISTI Innovations, which is earlier section 25, but not section 8 company, that means not for profit, which is promoting such kind of products. These are the formulas, different human formulations, which we have developed at the laboratory after validation, value additions. And these products are in the markets. These are some of the veterinary practices, particularly for the control of mastitis and then the ephemeral fevers. We are having almost all sorts of formulations, particularly for the animals, for the cattle, and even the birds. Even birds, to control particularly the protozoan and viral disease, we are having the formulations for that. Next. These are the regulatory aspects. Suppose we have come to develop the practices. Now, how to come into the market? That means we need to get the license. If we have identified the compound and done the multi-location field trial, then we may move for the CIV registration. But if we are not knowing, and the plant is only giving the plant growth promoting effect or stimulatory effect and having some pesticidal property also, then we may get the license in the name of G2 licensing, that is a biostimulant kind of things. So these are the ways, or even the plant, if the name of the plant is mentioned in the IPM, then we may get into the in the name of traditional formulations. So these are the three ways through which we can enter into market or categorize our product, either the IPM based formulations or the CIV based formulation, which is the mainstream formulation, or even the G2 license kind of licensing for the grassroots practices. Next. Yeah, this is a slightly deviation, but I would like to show you from the scientific point of view, because whatever the practices we are having in this history, that is our very primitive nature. We are calling it the informal knowledge, and this is our duty to after validation, we decipher the knowledge or the science behind that to put the information into the mainstream of the science. Here I am giving you one example for that, which work was done in the SISTI laboratory by a student from IIT Kharagpur day back in 2011-12, and that was the practices to control the blood clotting. The practice was people were using the spider web to inhibit the oozing of the blood from any cut and wound portions. So what we did, we have developed the cell line from the spiders, because if you go to literature, you will find it is the ampullate gland of the spider, which is secreting the mucilage for the production of or the saliva for the production of the thread. And that is being put, there are 4 such ampullate glands are found in the thoracic region of the spider. So what we did next, we have done the dissections. This is the area showing where you will find the ampullate glands and how it can be removed. And this is the detailed structure of the ampullate gland and the secretion of the thread by the spiders. So we have isolated the ampullate gland, and that we have cultured on the insect media, and even we have developed our own media to rear the ampullate gland. From that ampullate gland, we have developed the cell line, and from that cell line, we have isolated the proteins, because the thread is of proteinaceous nature. So we have isolated the protein, and that protein we have tested for the blood clotting in vitro condition. Next. So these are the how we have developed the cell lines. This is the picture of that in the plate. Next. This is the detail how the at what RPM and how much broth was taken for the formulations. You can read the things, but the important point in that we have done the, after developing the suspension culture of that particular cell, we have isolated the protein, determined the protein through Lorry method, the concentration of that, and that was formulated in saline, and then checked for the blood clot, done the blood clotting assay. Next. This is the plate and broth of particular this 0.4 mg per ml was the concentration what we have extracted, and this formulation 300 microliter of such formulation per 1.5 ml. Next. So this is the concentration. This is the amount of blood when we are adding 0.1 ml of the formulation in 1.5 ml. The clotting time was 3 minutes. When we have increased the concentration, clotting time has reduced, and when we have further increased, the clotting time was instant. The normal blood clotting time is 8 minutes. This is showing the positiveness, particularly the whatever the practice was telling. Practice was telling the use of spider thread to inhibit the blood clotting. And once we have developed the isolated the protein from the ampullate gland and developed the formulation, that is also giving the good result, and that was the concentration dependent. Means when we have increased the concentration, we have got the instant result. So this is showing the how we can decipher the science from a grassroots practices of just taking the spider web for blood clotting. We have come to the protein level for the formulation development. Even it can be accelerated on the larger scale or the pilot scale for the product development point of view. These are some of the insights. So, so the SISTI Labs has bridged many gaps. Many gaps means validated, and they came to know about the nature of the compound which is giving such effects for many such practices which are in the form of product now either at SISTI Lab or SISTI Innovations or even at the National Innovations Foundations. Next. So this was all about the journey of grassroots innovation, how we can validate, how we can develop the product, how we can add the value, or how ultimately we can decipher the science from that. If you people have some of the questions related to this or anything related to grassroots practices, you may ask.
Sir?
Yeah.
You had shown a slide where different extracts of plants were tested for their efficacy against pathogens and like against different pathogens, right? So In that, if we wanted to identify the compound which is responsible for activity,
Yeah.
would we be looking at something like a chromatographic analysis of those extra of the
Yeah, I
compounds?
told now HPTLC. Yeah.
Oh, okay. So would we be looking at compounds which are common to the more efficacious extracts, or would we be isolating each compound from each extract and testing it?
No, particularly it is based on the practices. Suppose the practices is talking about the sucking pest or chewing pest, or even for the controlling some pathogens. So which compound is doing that? After doing the fingerprinting, that is making the bands, we do the further assay to identify which band is giving activity, whatever it may be. So after assay, we come to know the number, depending upon the RF value of the band, which band is giving the efficacy. Once we will identify that, then we can tell that this band is having this property. Then the further we may even go for the mass and the IR for the particular compound identifications.
Sir, and one more question.
Yeah.
So if the practice involves something like heating, steaming, and things like that, how would we test the effects of those parts of the process?
I don't know, practice is for?
So if practice involves like steaming a paste or heating a paste or something of the sort, how would we test the effect? So the compound might have changed because of that, right? Or
Definitely. As so while we are standardizing the process means here in our case, from the validation point of view, not from the product development, from validation point of view, first the claim, how it has been processed by the innovator. Suppose if the innovator has done the cold extraction or the hot extraction, steaming, at how much times? Of course, the compound will nature will change, but if that is the process for the extraction of the particular compound which is giving the efficacy as per the innovator, we have to go for that. But at the same time, we also do the standard process of the extraction to know the different nature of the compound, whether such compound, suppose if the process is of steaming, we are doing even the cold extractions at room temperature, or even in the different solvent, whether we are getting similar bands in the particularly hot extraction or the steam extraction, or the extraction by means of autoclaving or sonications, or even whatever the current standard practices for the extraction each. So we can establish the fingerprint of different methods, and to find just to analyze those band pattern in different methods. But some of them would be of same, some of them will be of different. So even in our case, what we used to do that, if you do the extraction in similar in different polar and non-polar solvent, we generally enumerate the number of bands, 10 bands in one solvent, even 8 bands in, and their RF values. If it is same, then okay, same compound is present in different solvent. If it is unique in the, say for example, hot extraction, that means it is showing that it may be of one compound, but it would either degraded, which we cannot tell, because we will only come to this is the marker which may be having the active ingredient for that particular practices. Next step is to either elute the band or do the just assay by means of if it is related to some pathogens. As per the practices, we go further to identify its nature and the efficacy to establish the marker compound. Did you got or not?
Yes, sir, I understood. I have just one more question, if it's okay.
Yeah.
So the other question I wanted to ask was, when I was talking to Prof. Anil Gupta about my project, he suggested that I look at two plants, different practices, which could be affecting the same process, which is wound healing. So would it be okay if I looked at two different grassroots practices and tried to combine them? But I'm not sure how to go about identifying the basis of synergy of two different plants against wound healing when they've not been tested together.
See, if you see the, you may take more than one practices, it hardly matters. But the thing is that you first look from the literature point of view whether the plant is reported for that or not. That means you should be aware from the very beginning that whether you are taking a common practices or you are selecting a unique practices. You opt for the both. If you are selecting a common practices, you may add more than one plant which is having the synergistic effect. But if you are selecting a unique practices, we are not aware that two plant may have synergistic effect or may not. So we need to develop our design of experiment to do the assay for that. That is the thing. There is no harm in taking one practice, two practice. You can take a number of practices, but while you are designing the experiment, you need to be clear what you are or you want to, what kind of output you would like to have from your experiment. Accordingly, you can design the your plan of experiment.
Okay, sir. Thank you so much.
Yeah.
Any other questions?
Any other questions?
Any questions related to grassroots practices, method development, testing, everything, anything you can ask. Because evening you have a meeting with the discussion with professor, right? So before we can clear with Dr. Nirmal, he is a very good expertise in all those areas.
Everyone need to know how to get the practices from our available resources. Only then you will be able to plan some experimental experiment on that, whether it is either innovative or even the traditional, to develop the product based on that. As in the list I have shown you the at least 2000 practices available for different pathogen pests in the honeybee network. And if you see the literature, you would find that some of the plant is known for that particular purposes, some may not. So whatever is not known, that is ideal to develop a project for that.
If no more questions, then I would like to thank Dr. Nirmal for joining us today. Thank you so much, Doctor.
Okay, thank you. Thank you, Amar. Yeah, is there any question? No.
No question.
Okay, then. Thank you. Thank you. Yeah.
Thank you, Doctor. Students, please be with us. Our next guest lecturer will be joining us shortly.